Hi, everybody. Just a short update about what's going on. We're a public company. We are purifying our cells from the placenta after birth, so no ethical issues are related to that. And we are proud to say that what we found that the placenta is a rich source of cells, which enable us to do different products from the placenta, which I'll elaborate on that shortly. So we start with birth. 8 weeks after, we have the product cryopreserved in vial with a shelf life of about 3 years as of today. We expect to reach about 5 years, but we have to prove it in the ongoing testing, so we're quite confident that we'll reach it. Then we developed a dedicated thawing device to ensure that the quality of the cells will be kept after thawing to ensure proper injection of proper cells. And we are moving forward in a variety of clinical studies. The most advanced program is CLI, critical limb ischemia. You've probably heard from other people too, become very popular in recent days. We have been approved for phase 3 by the FDA, which we intend to start shortly. We're waiting for the European agency approval of that study, so it will be a multinational study, multinational phase 3 study running in parallel in US and in Europe. About 250 patients with the endpoint of amputation-free survival. In Japan, we have been approved under the conditional approval, 75 patients, which will be run in a separate study with a little bit slight different endpoint. And we hope that in the end of the day, we'll reach global coverage of that indication in the world. Okay. The second indication is from the orthopedic arena, femoral neck fractures. We demonstrated good efficacy in elective hip fraction operation, and we made a decision to move into the femoral neck fraction because we feel that mainly due to the elderly population, there is a need for such an indication. We are in a process to negotiate an SPA with the FDA and the European agencies. And then upon approval, we'll start phase 3 study in that indication. Everything is based on the phase 2 data that have been presented more than a year ago, where we demonstrated that with IM injection of the cells into patient immediately after the operation, we reach 500% improvement compared to the placebo in the muscle force, 300% improvement in the muscle volume using MRI, and an interesting phenomena that on the left you can see the operated limb, and on the right is the contralateral limb, and you see that there is a significant improvement in both two legs, suggesting that intramuscular administration can generate systemic effect. The third program is R18. R18 will be used for hematological disorders. The product is sponsored by the American NIAID with the goal to use the product for acute radiation syndrome and to stockpile it in case of catastrophe. The product is now under large animal studies. The product should be approved by using the animal rule, which is accepted by the FDA. So we expect data in the next few months. And then, of course, we'll move forward into a pivotal study that will approve the product under those guidelines. We have, in addition to that, we have cooperation with Fukushima Medical Center to understand the use of our cells for treatment of not only for hematological ARS, but for GI and lung ARS, which is needed in Japan. You can see here additional studies that we are running in parallel. The IC study is 107, intermediate education study is running in parallel in US, Europe, Korea, and Israel. We're about to finish the study, it's 170 patients. We hope to complete enrollment in the next few weeks. And then we have been approved for R18 study for insufficient engraftment to inject our cells following failure in bone marrow transplantation, and the study is ongoing. You can see here our manufacturing facility. We have been listening to the finding of our industry that in cell therapy, the process is a product. Luckily, we discovered that phenomena 10 years ago, and we invested heavily in full automation to have a full automatic manufacturing process that can produce hundreds of thousands of doses from a small facility. The facility is in Haifa, Israel, have been approved by many agencies, FDA, EMA, Japanese, PMDA, Korean, Australian, and inspected by the European agencies. So we are proud to say that our production line is ready for marketing, not only for phase 3 study, which is unique in our space. A few words about the uniqueness of our platform when we combine the placenta with manufacturing. First of all, the placenta is a rich source which enables us to have different products. I described to you 2 products. They have PLX-PAD and PLX-R18. We have additional products under development. All of them are from the placenta. The placenta represents unique immunological properties. You probably hear it from everybody in our space, but the only test to claim that the cells are really immune privileged is to inject twice from the same donor to the same patient and to look into the immunological characteristic or immunological interaction between the cells and the patient. And we demonstrated that 2 injections of PLX cells from the same donor to the same patient, 2 weeks apart, 2 months apart, 3 months apart, does not generate immune response against the second injection. And that's the only evidence that's really unique from the immune prevalence point of view. We think that we understand the mechanism of action. You have heard from many people about the complexity of MSCs. So we invest heavily in the last few years to understand the mechanism of action. And probably the best hint is to Alain. When we had some communication years ago, the first question that I've been asked by him, can you explain the mechanism of action? On that time, it was complicated, but we took the challenge and we invest heavily into that understanding. We published a few articles on that. So we think we understand better how to target the product to the indication. We even demonstrated that when you take different products, for example, our PLX-PAD and PLX-R18, in the same animal models, you can see different response because the cells secrete different cocktail of proteins, and I don't think that anybody demonstrated it yet. I think that our claim today, which is supported by the understanding of the mechanism of action, that cells are not identical. For each indication, you have to have the right cells that secrete the right composition of proteins to do the therapeutic effect. And the only proof for that is the phase 3 study that we are running now. Manufacturing is key, as everybody said before. You've seen the picture. We think that even in the immune therapy, people realize today that in cell therapy, without good manufacturing, nothing will be developed. So we are proud on that, and we are investing ongoing on that issue. And of course, as we say all the time, at the end of the day, the project is finished when the product is approved. So we are proud about Argenic that is in the process of approving the first product. We hope to be the second, and we are moving forward toward that direction. That's my presentation. Any questions, any issues? We have a few minutes. Okay.